Hello, welcome to today's Catana tutorial, which will focus on loading and visualization of oxDNA structures. Since Catana does not offer direct import of oxDNA files, we will use Unified Nanotechnology Format as an intermediary between the oxDNA or oxview and Catana. This format is freely available together with its description, converters, viewer, and so on, and is supported currently by both Catana and oxview tool, with support of other tools coming soon. So let's start. First, select the structure we want to load. In this case, we will go with the tetrahedral protein cage structure from the Nanobase server. So first, download the oxDNA topology file and corresponding oxDNA structure file, and head to oxview. In oxview, click on the open file and load these two files and wait a while. So you see the structure was loaded. So now we can take a look in oxview on our structure and export it to UNF. To do so, click on the UNF export in the top bar. As you can see, the structure was exported. So now we can head to Catana, go to structure import, and drag and drop this UNF file to the corresponding place. So now again you can perceive the structure has been loaded to Catana. So now, as you can see, it's the same structure, just in different visual representation, and we can play with it or change the representations. So let's start. If you perceive the structure now, you can see that we have some DNA nucleotides, some amino acids, and we have some chains and some IDs. So the numbers correspond to the global IDs of the structures in the UNF file. So we can use these IDs now to filter out the structure to provide some more visually pleasing outcome. So let's start by adding tube representation and visualizing all the monomers, which means amino acid or nucleotides, which are coming from protein or have ID not between 0 and 16,000, because if we perceive there are IDs going to 16,000. And now we can add double-stranded representation, which will take some time because the double-stranded representation tries to compute base pairs, which means basically compute the positions where are base pairs to provide more abstracted representation of the structure. In this case, we will visualize only monomers with ID between 0 and 16,000 and also only DNA parts. So now, as you can see, we have the double strand, let's say in this cage structure where we emphasize the double strands. And then we have the tube representation for DNAs, which are connecting the protein parts. And then the tube is also used for the protein. To further emphasize the protein, we will go to this custom color filtering and make the protein parts, let's select yellow as emphasize them enough. Then we will make the DNA parts, you can make them white, or let's make them somehow green, or maybe blue. So now basically we emphasize the parts which are connecting the cage structure to the protein. As you can see, sometimes there are positions where there is only a sphere. It means that in this case the Catana was not able to match corresponding base pair. So these are either single-stranded nucleotides, or let's say single nucleotides, or it could be base paired nucleotides which are not, let's say, close enough for Catana to identify them as base pair. So this is just for the information. In any case, let's say we adjusted this representation in a particular way. We can also change the coloring scheme based on our preferred one. So we can maybe focus on this crossover one. And now, let's say if we have some representation we like or we prefer, then we can, for example, toggle, or we can modify the fog settings, so we can make the things which are far apart disappear. And now we can head back we can head to screenshot export, structure export screenshot, and preview it. Say, okay, this looks good. So we can upscale the screenshot, trim the empty space, keep the black background. And now we can see that we have nice screenshot we can either download and perceive. We can also download the screenshot with transparent background. So if you will generate it, now it will have a transparent background. And we can use this screenshot, for example, somewhere in a publication. And of course, there are more things we can do with the structure. We can modify it. We can duplicate the structure. We can extend some strands. Or we can delete the representation, all of them. And again, the tube representation, I visualize everything in tubes, for example, except the proteins being red. Yeah. So there are multiple things we can perform, and everything is highly modifiable in Catana. So feel free to play with the structures. So that's it. And final thing, that now if you would modify the structure or perform any operations, we can also export it back as UNF file and import this file in oxView, and then, for example, go through oxDNA simulation again with our modified structure. So that's it for today, and thank you for watching this tutorial, and have a nice day.